Thank you, Moritz, so much for the introduction and for inviting me to be here. Thank you, all of you guys, for attending, and for those of you who have presented already. This has been the most densely enjoyable forum, conference, symposium, gathering that I've ever been to. So this is just extremely energizing for me. So my training is initially actually in developmental biology and cell biology, but my focus, my interest is really, I think, quite closely aligned with a lot of the folks who are here today, which is in understanding diversity, understanding the diverse world around us. And one of the tools that I use for that is to study the dynamics of cells, the dynamics of how cells are changing their shapes and how they collaborate together to generate whole tissue and whole body forms. And so that's why I've put these things up here. These are some samples from some of my research that I'm actually not going to talk about today, but I put them up here as a reminder to myself and to you guys that the cells I'm going to be talking about today are incredibly dynamic. And what I'm going to be talking about instead are snapshots of that dynamic stuff that they do. I'm sorry, I forget his name, but the speaker who spoke just before me called cells these tiny, beautiful things, which is, I think, going to be a nice way to summarize what I'm going to be talking about today. So the thing that I think unites us conceptually is this interest in quantifying tissue traits. And I spend some time on this live imaging and live dynamics, and I spend some other time building things to manipulate tissues in physical ways. And I won't talk about that today, but if you're interested in talking about those things, I'd be happy to chat later. So instead, here is the plan for today. First, I'm going to suggest a particular way to use cell and developmental data to inform our understanding of ecology and evolution. Then I'm going to make the case for using existing published images from journals, right, as data sets. Then I'm going to talk through one example of this in detail. And finally, I'm going to, if I have time, touch on another example very briefly. And this is basically going to have the rhythm of short, long, short, so to speak. I'm going to hopefully reserve a lot of time for questions at the end. So please feel free to ask lots. So there's an incredible amount of diversity and disparity in life today, and all of that is just a tiny sliver of the diversity that has existed throughout the history of life on Earth. It's a great quote. I couldn't have said it better myself, and so I'm not going to try to. I'm just going to repeat it, and I checked with my source as a good journalist would. So yeah, I mean, we all know this, right? There's like this insane amount of morphological diversity for us to understand, and it's actually worse or better, depending, than it would be if you just show a slide of diversity. So I'm actually not going to show a slide. I'd rather you guys conjure in your minds whatever, clade or taxon that you particularly like to spend your time focusing on, and then notice how, as you learned more about it, you've learned that the problem is actually harder than you thought it was at the beginning. That as we zoom in closer, there's this like fractally expanding set of details or of stamps to collect, depending on who you're listening to. And we have this big bifurcating process that generates all of these data. And what that means, in a sense, is that actually every morphological object that we study is unique. It has its own particular evolutionary and ecological context, and it's got all these contingent events in its past. So we're seeing a snapshot through time. And the fact that everything is unique is two things. On a good day, it's good news, because it means there's this limitless set of stuff for us to understand. And on a bad day, it fills me with despair, this like existential, epistemological ennui that there is so much out there to understand, and it might literally be impossible to make sense of some of the morphological diversity that's out there. Like it might be, information theoretically impossible for us to make sense of some of those things. And I think we should always keep that in mind as a possibility for some of our questions. Ask, can we make sense in an orderly way of these data? And then if so, what are those orderly patterns? All right, so that's what I'm going to try to get at for a second here, which is, are there generalizable patterns of morphological evolution? And if so, what are they? Okay, so I tend to be sort of strict on what counts for me. I think one thing that definitely counts is these broad scaling relationships across multicellular organisms that emerge from sort of fundamental physical limitations of how things diffuse and of how temperature affects the speed of vibrating molecules. I think that's a pretty good case, and we've spent lots of ink on that over the last 50 years or so. I'm going to propose a potential additional case, which is that broad patterns could emerge from the fact that all bodies are built from the same relatively small set of highly conserved cellular structures and behaviors. So, okay, what do I mean by that? What I really mean is that for at least multicellular organisms, you can characterize their features in this gigundo, high-dimensional space. And that if we zoom down to the cellular level, we might be able to extract low-dimensional features from a smaller set of these basic conserved things that cells do. So this is an idea that I'm really proposing for you guys that has captured me, and I think it could be useful for us, but I'm also quite willing and eager to hear pushback on that later if you'd like. So here's this idea that Megan and Masahito both talked about yesterday, this idea that genotype and evolution give rise to a phenotype. And I think, in my mind, I tend to reconceptualize this very slightly as this function, right? There's this function that takes genotype and environment as inputs and generates a phenotype. And this function, of course, anybody? It's development, right? So development, really, it's just this process, right? And in multicellular organisms, it's a process that generates a particular spatio-temporal arrangement of cell numbers, cell sizes, cell shapes, cell packing, and cell secretion. So, I'm coarse graining, an entire field here, and you might be thinking, wait, hold on a second. I've taken cell biology before, and cells are complicated, right? So yes, I'll be the first to admit that cells are themselves extremely complicated on the inside, and they interact with each other in lots of complicated ways, and the textbooks are therefore very heavy, right? But I'm going to suggest that we have a useful handhold here, because cells only evolved once, and we have this unit in the cell. We have a pretty convenient place to coarse grain the physical world of organisms. And so I'm going to suggest that we do that as a way to help making sense of the diversity we see around us. And when we do that, then morphological evolution is just the change in cell number, change in cell size, change in cell shape, and packing and secretion. Okay, so I've done a little bit of rhetorical jujitsu. And I started from, can we make any sense of this morphological diversity in the world? And I've kind of rearranged that into this narrower and ideally slightly more tractable problem, which is, how do changes in basic cell processes give rise to diverse tissues? Okay? So to answer this question, we need to sample cellular data from many species, if we want to understand these at a broad level, right? However, cell-scale morphological data are very labor-intensive to collect. Right? They're so labor-intensive to collect that, in fact, you typically have a single lab working on a single tissue, working in a single species for the entire lifespan of that lab, right? So it's difficult enough to collect this data from just one species or a handful of species. This is a reason why the Evo-Devo conferences tend to be like, we studied one new animal. That's important, and that's what my whole PhD was about. It's really good, right? But we would like to be able to sample more broadly, especially if we want to understand macroevolutionary patterns. It's just typically intractable to do this for hundreds or thousands of species at a time. But fortunately, there's a little trick that nature has pulled for us, right? Which is that it can be more tractable if we collect that cell scale data by observing the record that the cells left behind, which is the extracellular matrix. Okay, I don't know how long it's been since you took cell biology, developmental biology. The extracellular matrix is what we see when we look at each other. We're not looking at cells, we're looking at the stuff that our cells have secreted, in this case on the outside, right? And extracellular matrices are actually just all around us, right? So I actually work on the development of all of these species here, but I'm mainly showing them here because everything you see on them actually is, on the outer surface of these animals, is an extracellular matrix, also ECM for short, which I'll probably use interchangeably. And in this case, actually what you're looking at are so-called apical ECMs, which are materials that are secreted from the apical surface of epithelia specifically. So these ECMs do many important things, tons of important things actually, and there's whole symposia devoted to this in other places. They hold the tissues together, they mediate the physiology of the tissue, they protect you from the environment, they can influence how morphogens are actually secreted and moved around in a body, and then they serve as the substrate for other moving cells. They walk on each other, typically by walking on the ECM. But also, and I should say, because of all this stuff, it's just intrinsically valuable, of course, to study ECMs. In my case, I think it's also just useful to study ECMs as a tool to make inferences about the shapes and behaviors of the cells that were there during development and over the course of evolution. Okay, so I do this with one of my study tissues, which Moritz mentioned, the insect egg. so cool. All right, so there are some important things you need to know about insect eggs before I start. And the first thing is that eggs are delicious. Just so yummy. I love eating eggs. In my case, it's chicken eggs and fish eggs. But I'm in good company with a lot of species out there who just love eating eggs. And all of us together, me and the egg parasitoids and all the other egg predators out there, we like to eat eggs because an egg is full of energy and nutrients, right? It's everything you need to grow an entire organism, right? And that's the function of the egg, right? It's a container of goodies to make an entire organism. And because it's got everything you need to make an entire organism, they're a nutritively rich target. And they also, you'll notice, cannot run away or hide. So they have this eggshell on the outside to encase and protect those goods. Also, the embryo that's developing inside, it needs to metabolize. It needs the right amount of oxygen and water. So the eggshell mediates gas and water exchange to that developing embryo inside. And finally, this is the thing I'm going to spend most of my time talking about. These eggshells are super duper diverse. So maybe I'll get a drink of water, and I'll pause right there if anyone has any questions about the intro, and then I'm going to pivot. Okay. All right, so before I go on, I'm going to give a little primer on how eggs are made, and how eggshells specifically are made. All right, so each egg forms in this organ-like structure called an egg chamber. This is in insects, by the way, although there's actually some very similar analogs, not homologs, analogs in other animals. Okay, so the outside of this thing is a continuous sheet of cells, an epithelium in magenta. It's called the follicular epithelium. And it surrounds and supports this orange thing, this yellow thing in the middle called the oocyte, right? So that's the thing that's going to go on to become the mature egg. And this epithelium on the outside is really my object of study. But many of the ways I'm going to learn about this epithelium is by studying the shape and size and the surface of the egg that it eventually produces. Okay, so I'm going to zoom in a little bit, just very briefly, onto one cell. And it's the apical surface of this cell that faces the oocyte. So this apical surface is facing up. Sometimes we think of this as the upside of cells, in which case the upside is facing inwards. And it secretes this eggshell ECM onto the oocyte, apically. And then this epithelium is shed, and what's left behind is this structure over here that's surrounding the mature oocyte, okay? And often, the eggshell ECM retains footprints of those epithelial cells that were overlying it. Now, if you study something like Drosophila or certain other species, you might notice that there are some peripheral structures that I've omitted here. I'm basically showing the parts of the egg that are shared across all insects. Okay, so, with this structure in hand, we can now study variation in the egg and in the tissue that makes it and in its eggshell at 3 different scales. At the tissue scale, the whole egg, at the cell scale, or at the subcellular scale. And I'm going to tell you some results about all of those today. And on that note, I'll say, this is not a methods talk. I've tried to actually go pretty thin on methods because I'm not really a maker of computer vision or machine learning methods. I'm just an enthusiastic user. So I figured it could be useful for us to talk through an example case and show how these methods get used. And if you have any questions about that, feel free to pipe up later. Okay, so I began by just collecting some papers on insect eggshells, right? And these are the experts who already knew about insects and how to identify them as species, how to get their eggs, not always easy. And they made some scanning electron micrographs of them. And the thing you learn is that cell imprints are present in many groups of insects. These are actually quite distantly related insects. And also, cell imprints are absent in lots of groups too. So I was like, all right, time to start getting some data. So I set out to collect these micrographs systematically. I was reading a book recently where a character said that the love of knowledge is a type of madness. And I was like, it's set in, because I decided that's it. I'm going to get every micrograph of an insect egg that has ever been published by a human. I set out to do that. And at the beginning, I had no particular tools to do this. And so it was a lot of manual work. And then along the way, you start trying to automate things. You start writing scripts. You start coming up with rules. And eventually, you sort of systematize this process so that it becomes a little more reproducible, ideally. And then, even better, extensible to other tissues and to other people. So along the way, though, you end up going down some little side roads, some little rabbit holes. One of those rabbit holes I thought I'd mention right now, which is I found what I think is the first scientific description of an insect egg by Jan Swammerdam, who, when he was published in England, eventually was renamed John, that's how they do that. And this is an image of a butterfly egg. He actually did a lot of this work when he was like exactly my age, which is kind of annoying. He just wrote this like 400-page book. Tragically died fairly young. This was published posthumously. He actually also has the first observation of red blood cells with a microscope. He observed insect oogenesis for the first time in detail and describes it. It's wonderful. Anyway, I didn't use this. I only used actual micrographs, even though there are some beautiful hand drawings of tissues. I limited myself to the micrograph. I figured there's already enough human intervention in the data. Let's not add more where we don't need it in this case. Okay, so my collaborators and I assembled this set of PDFs with digitized text, and in many cases we had to do our own optical character recognition on lots of older documents. But this is one such example. And then it's important to note, right, that I want the images of eggs. That's what I really care about. But there's also essential information in the text itself, or as you might put it, the labels, right? So here's an example. We have a genus and a species. Definitely need that. Definitely genus and species. And we also have some morphological data on the egg, the length and width of the egg. And then you might also notice that there's some actual textual description of what the surface of the eggshell looks like. The exochorion is just the outermost layer of the eggshell. I did not use this. I have it, but I didn't use it. I set it aside. I figured I'm just going to take the images themselves. And we also wrote some tools to automatically check for completeness. And you don't really think about this at the start of a project, but a fairly large problem was taxonomic errors and synonyms, because we're renaming creatures over time. And this is actually, and there's also just errors in the published papers too. There's typos, there's things misnamed, there's like bizarre numbers that don't make any sense. So you actually have to do a fair amount of cleaning that's just due to the human input to the data. Wrote some tools for that too. So you can imagine this whole process, coming up with a tissue of interest. In this case, it's this conserved structure. It's the egg, and it's the eggshell. You can describe that with some text strings. Eggshell, chorion, egg. Then you have some other things that we want, which are the taxa, insect, Lepidoptera, Heliconius, and you have those strings. And what we're really just doing is taking those strings and combining them in a certain set of permutations and putting them into a database, and then retrieving a bunch of links. Those links bring you down to PDFs. PDFs have extractable text and images. What I'm starting to describe here sounds like something that's a little bit boring for a human, but a little bit fun for a foundation model, right? Like ChatGPT loves this stuff. You could imagine making a tool where you build on these things to extract the text in sort of a structured, reproducible way. You give it instructions, it proposes, and you refine search queries. Then you can use a database, pull down these papers, do optical character recognition where you need to, associate text labels with images. Turns out this is a problem that has also recently been solved, helped by the fact that on arXiv, you upload the raw LaTeX file, and you also get a PDF. So you can associate the spatial information between the text and the actual figure that's matched to it. So you can use it as a training set. It's very cool. And then you come up with basically candidate data points, which is like an image and the text associated with it. And you want, this is a fundamentally human enterprise that we do here. So you want a human to see whether this is in fact the kind of thing that satisfies their curiosity. I think at some level, depending on your interest, you might want to do just spot checks. In this case, we didn't do that. We did human eyes on everything. Anyway, you get this really nice tool. And you can import this tool. And there's one problem. Actually, I never got around to writing this tool. So this tool doesn't exist. But I really think it's possible. And in fact, I think all the components of this completely exist right now. And one of you, if you want to make it, certainly could. And I have suggestions. So it's actually not a tool. This is really a proposed tool to do this. And I think it would be really great. And I think it's actually inevitable that sooner or later, how many papers do you guys read per year, right? Compared to the number of papers that are published. These lines are diverging, right? So we're going to need solutions. And it's not entirely obvious to me that using prose is actually going to be the most efficient way for us to communicate scientific findings like 30 years from now. But in the meantime, and the fact that we have these tools that eat and poop prose, it's actually a fairly reasonable thing to do. So I actually didn't do this. Instead, I had a lot of little bespoke solutions that made my life a little easier. But in fact, this is mostly manual. All right, so here's some of the results. These are eggs. This is a tiny little subset of the data, but it's this big corpus of micrographs. And entomologists have long suspected and proposed and spent a long time talking about how some of these features, these really interesting, complicated textual features, might make sense for the functional context of the eggs and their ecological niches. Predators, parasitoids, whether you're in water-rich or water-poor environments, all that stuff. These have not really been tested very well, but that's what most of the literature is about, in addition to some of this just pure descriptive work for taxonomic purposes. So I just treated this as a big data set. And I worked on this with some collaborators who I've been working with for a while, Sam Church and Bruno de Medeiros. And now we've got this big data set. Now we can start asking evolutionary questions. All right? So for instance, let's just reconstruct the tree of cell traits, right? And in particular, just are there cell imprints or not? Right? So I'm going to show you the result of that. I'm going to show some branches that are red that show you those branches where we've done ancestral state reconstruction, and they do have cell imprints, and in gray are those that don't. Here's the data. Every one of those tips is actually not a species. That is a genus. So we have a lot of data here. In fact, in some cases, we have many individuals per genus, but our tree resolution only goes down to the genus. We ended up having to make, actually, an updated insect phylogeny to handle these data. And I'll just zoom in a little bit to show you some interesting features. One thing is that the last common ancestor of insects likely had smooth eggs. That's a robust, high-confidence finding from the ancestral state reconstruction. Also, there have been a lot of independent gains and losses of this trait, which is interesting. So here you can see some independent gains in the tree. In another part of the tree, in the holometabole, you can see several independent losses. And in fact, very conservatively, at least a dozen independent gains and losses of this trait. And then, of course, with these images, I'm sure you can guess where this is going. You could use image analysis tools to measure a bunch of tissue features. I'm not going to talk about the details of this, but I think it's pretty straightforward to imagine what you would do with these. You could take these images, you can segment them, and in this case I'm using, the best one for me is this thing called cell pose, which is basically a zoo situation where they have trained on a bunch of diverse samples, almost none of which I think are actually scanning electron micrographs. Mostly they're fluorescence images and transmitted light images. But because of the generalizable features of how closed shapes look in tissues, it has learned to recognize celly things. And those cell-like things just work quite well on these data, which is nice. So then we can infer cellular traits on the basis of the imprints that the cells left behind. So we can infer cell number, cell size, shape, packing, secretion. You guys remember this list? Okay, so this has resulted in this big multi-dimensional data set, roughly 10 traits per imprint, hundreds of cells per species in many cases, and roughly 1,000 species. And there's representatives of all the insect orders. And so, it's useful to think about what this data set can and can't give us. Typically, there's just one tissue per species, right? People only publish one tissue per species. So for this particular data set, it's a point estimate, basically. But of that point estimate, you do get distributions of cell traits within the tissue. So here's just 3 example tissues and the inferred cell imprints, and then here I'm just showing you the distributions of aspect ratio, one of many things you can measure about cells. And then with these data in hand, we can start to address questions that people have bandied about for a long time, but really haven't had the data to ask. So how does cell size covary with tissue size? Right? It's a pretty simple question. And before I tell you the results, I should just point out that the whole tissue varies quite dramatically in size and in shape. So it's this tissue, this follicular epithelium, is homologous across insects. It's highly conserved. It's present in every insect we've ever looked at. It also has a really broad range of shapes and sizes. So here's the size data. I'm going to show you, this is one of the smallest eggs that's been recorded, this parasitoid wasp. And I'm going to show you only egg silhouettes from other species within Hymenoptera, which is this clade that ants, bees, and wasps. That guy lays, or she lays eggs in other eggs, so your egg has to be smaller than the egg that it goes into, so that's one constraint on you. But there are larger eggs too among ants, bees, and wasps. And in fact, some of them are a little more regular size, some of them are a little bigger, some of them are even quite large. In fact, the biggest egg here, this is the longest egg we have in our data set, this carpenter bee, it lays an egg that is longer than the shortest hummingbird egg. So these are all in the same subclade of insects. And anyway, so now back to our question. How does cell size covary with tissue size? And this is this long-standing question. It's essentially an allometry question. And what we really want to know is, over many independent evolutionary events, what are the general tendencies in these cell traits and the tissue traits? All right, so here's some of the results. So what I'm showing you here is on the y-axis, that's a representation of tissue size, the surface area of the ultimate egg, which is almost a perfect match to the surface area of the tissue that was secreting the egg, right? And then the x-axis represents cell size, here measured also as an area, the area of the cell imprint. And each color here represents a group of insects that had a single common ancestor. So these are all clades within insects. And note also that both of these axes are on a log scale. So there's actually a tremendous amount of diversity here. So for a given egg surface area, you can actually get a 300-fold range of cell size that generates those tissues. And also, I should point out, each dot represents a whole genus. Okay? So the main answer to this question really is looking at this is that actually there's different rules for different clades. For instance, in butterflies and moths, right, shown in light blue here, they're predominantly varying in cell size and less in tissue size. Flies and mosquitoes are the opposite, right? They're predominantly varying in tissue size and comparatively less in cell size. So to put this in the framework that we started with, if we think about the evolution of morphogenesis as a limited set of knobs, oh yeah, you have a question.
Yeah, I have a question. So you said each of these dots represents a genus, but like, within a genus, do you have pretty like narrow bands of what the varieties would be?
Great question. By and large, yes. It's more the exception than the rule that there's a lot of variation at the genus level. And there's probably like 6 cases where there's dramatic change at the genus level, and I keep a close record of those because if I ever start a lab, that's like what we'll work on, because you want to be able to do some crosses and that kind of thing. By and large, within a genus, the traits that I'm showing you here, those dots would like stack on top of each other on these giant log plots. Yeah, good question. Yeah. So, all right, in this big framework, there's a few things. I'm proposing that you might conceptualize development as a few knobs. And if there's a few knobs that are being tuned over the course of evolution, this suggests that in some of the knobs sometimes get a little bit sticky in certain clades, or more sticky than in others. Or maybe in certain clades, actually like your toddler came over and like pulled that knob off, and certain things like stop changing altogether in certain clades. This is the case, for instance, in the ovarial number within ovaries. So here's another question we can address. When a tissue evolves a different shape, how do the shapes of those cells change? And again, I'm just going to give you a little primer on the shapes of these tissues. So that's a kind of a typical looking insect silhouette. This is Drosophila melanogaster. You do get eggs that are appreciably longer and skinnier, and others that are appreciably more squashed, right? I call this large aspect ratio, small aspect ratio. Or we also see there's some that are more asymmetric, others that are more symmetric. And I'm leaving implicit here and not going into detail about this, but there's actually a known homologous axis across all these eggs. It's basically an axis of rotational symmetry that's this way for all the images I'm showing. So there's actually not quite as many degrees of freedom for eggs as you might guess from the silhouettes. But we can talk about that more later if you're curious. So just to show you, here's actually the data we have on egg size and shape. So size is shown here as volume on the y-axis, and aspect ratio is basically the first principal component of shape shown on the x-axis. And each color, again, is a clade within insects. And what you can see is that volume-wise, they span about 8 orders of magnitude, but also the shapes span quite a range. And in fact, if you kind of scroll through a couple different clades, and just kind of blur your eyes and look where these points start landing for different clades, you'll notice that the points of different colors overlap quite a lot. And everywhere you see those points of 2 different colors overlapping, that is convergence in morphospace, in this morphospace. So the rough way to think about this is that actually tissue-wise, convergence is rampant for this structure. And this means that evolution basically ran this morphogenetic experiment for us, and fortunately it used an experimental setup with a lot of statistical power. Okay, great. Unlike most of us, right? Just kidding. Okay, so when a tissue evolves a different shape, how do the shapes of cells change? Again, this is a question of evolutionary allometry, not static allometry or ontogenetic allometry. And this I'll go through sort of briefly because it's kind of a simple summary. This is a subset of the data for clarity. But the basic answer is actually the cell and tissue shape do not covary in any predictable way once you account for evolutionary relatedness. I found this completely shocking, and this is actually counter to the hypotheses in the literature. It seems like, of course, yeah, you change the shape of your tissue, and the shapes of the cells are going to have to move with that, but that's not true. Okay. Another question is, under what circumstance does an epithelium evolve a radically different cell packing arrangement? This is an important topic for people who study the mechanics of tissues. And so if you look at these guys, this arrangement on the left is what you might think of as sort of a kind of a typical epithelium. They tend to be roughly hexagonal. There's actually a good deal of biophysical and theoretical reasons to expect this sort of distribution in a world where there is some tension on the edges, or in a world where you're sort of limiting the edges, or you just distribute your neighbors after each division in a certain way. This is roughly what we expect for a typical epithelium. Then there's these other bizarre arrangements that are found almost nowhere else in nature. In fact, I've never found another triangular tissue anywhere in nature, and this sort of rectilinear grid, again, very rare, only transient in the development of certain species have we seen that kind of arrangement, unless you're aware of any others you can tell me about. I find these completely amazing, and as I was poring over the source images for this, I was sort of doing a little, binning in my head. I just call these like, mostly hexagons, mostly rectangles, mostly triangles, and I'm like, all right, I'll come back to that and actually figure out what the real dimensions are of this. Like, let's figure out how they really cluster. So I did some more complicated things. You can characterize the topological arrangement of these cells. They're packing in lots of different ways. You can characterize their geometric features. And then you can, here's one example. This is a measure of rectangularity, which is sort of an intuitive concept to humans, a little bit challenging to think about ways to actually describe it in math. But this is just one of many metrics. And so you take all these topology and geometry measures, and then you just basically look at all those data, all those traits, and you see how they tend to cluster. And it turns out, to my astonishment and chagrin, that those first three bins are actually, these are the clusters actually. They do cluster into these bins. I don't know why, and I think it's actually kind of conspicuous and interesting that they do. And it's actually, honestly, hard to overstate how weird these arrangements are for developmental biologists. In fact, I present on this at developmental biology conferences, and every time there's someone who's like, those are not cell arrangements. I promise you, that's not possible. And so, I was actually fairly skeptical myself, so I had to do some time to figure out if this is really true. So one thing you can do is, you can ask, wait, do these polygonal patterns really represent the cell arrangements? Okay, the short answer is, in every case that's been checked, the answer is yes. And it's only been checked in a small number of cases because it's so labor intensive to dissect out a tissue and look at where its cells are. But here's an example from a pea-eyed butterfly that, you have this rectilinear arrangement of cells, and you have a rectilinear arrangement of cell imprints, and they match. And then also in these Pyralidae, these are basically pest moths that lay eggs inside your pantry that will eat your dried foods. They have a lot of these sort of triangular arrangements, or these like lens-like arrangements where you have 2 neighbors per cell. Turns out that, in fact, when you look at the cells, they have that arrangement in this tissue. And just to make it extra clear, I decided to check this myself. So I started raising these guys in the lab. These are these moths, Plodia moths. They're really easy to rear, and they lay infinity eggs. And I just popped this onto an environmental SEM. Turns out the SEMs are so good these days. There's no fixation, no sputter coding, nothing. I just put this in, and 5 minutes later I had this image. And you can zoom like way into like the nanoscale. It's amazing. And then, so this is the egg. In fact, it's a good representation of the egg. I can check that that's what the egg looks like. And then I also dissected out some of these developing egg chambers in the ovary. And in fact, they are triangular cells in there. And in fact, they're so tightly triangular that they squish the nuclei, and the nuclei are triangles too, because they are squished down by the cell-cell interfaces, which is also bizarre, but real. Okay, so the question, of course, right, is how has this evolved? What is going on here? Are these some weird evolutionary one-offs that have shown up for some bizarre hiccup that happened over developmental history? It also asks a lot of questions about the molecular control of cell alignment, which I think you would really want to study in a species like this. That's something I'm in the process of doing. So one question, of course, how does epithelial cell packing evolve? And really, are these unusual one-offs or not? So to answer this, of course, we just did ancestral state reconstruction, which allows us to infer changes in these traits over time. Here are the results. Okay, so it turns out, actually, the answer is just a no. They're not one-offs. So triangular and rectangular packing have both independently evolved multiple times. This is actually a downsampling of my results to make it visibly clear. In fact, this is actually underestimating the number of independent changes that have probably happened for these traits. So I think, these arrangements are really far from what you might think of as energetic equilibrium. And I think it's going to be really interesting to figure out how these are formed and how they're maintained in the developing tissue. But that's not really the point of this talk. Okay, so I'm going to talk about one last trait, which is the pattern of surface heights. This has also changed dramatically. So I could give you a bunch of, I call this sometimes the topography, or think of these as little landscapes on the eggs. And the sizes and arrangements of the cells in this case are almost identical, but what's changing is the height of where this extracellular matrix is being deposited and maintained. really different and really interesting, and this tends to be a very evolutionarily labile trait. It also allows us to infer some things about how the cells know directions and polarity within the cells. Okay, I can talk more about that later if you want. I'm gonna move on though. So here's what we have. We have this tissue scale, cell scale, and subcellular morphological data. Lots of dimensions of variability, hopefully fewer than those that we see out in the world for all tissues, but we're going to try to get all the cell data we can. And this raises some questions. Namely, one, do ecological traits help to explain these patterns of cellular diversity? And two, what are the mechanistic bases of how these traits vary? What are, in fact, the meaningful knobs that cells are tuning? So for the sake of time, I'm just going to answer one of these right now. I'm going to focus on this one here, the mechanistic basis. Figure maybe some folks from around the department maybe study development, they might be interested in this. So it won't surprise you to know that the mechanisms underlying eggshell formation are actually experimentally tractable in Drosophila. So a lot of my work I actually do on Drosophila. And here's an example of what that Drosophila eggshell looks like. A little bit simpler than some of the ones I was showing you, but also just amazing. The cells are gone, but they leave behind these high fidelity imprints. So we can learn aspects of their secretion and their morphology. And for this portion of my talk, I'm collaborating with a few folks in the Horn-Badavinnik lab, shown here. And what I'm going to do now is just very briefly tell you about a few transgenic tools that enable us to study this ECM in closer detail, and then I'll show you a few quick results that we can learn from those. Okay, so I'm going to just remind you that eggshell is secreted onto the oocyte, and it's secreted in layers. So now I'm going to decompose these into two layers. The inner layer has these pillars, like pillars in a building. And then on top of that, there's a roof, right? And these pillars have some orderly spacing, which is, people who are used to looking at textured images, there's a really conspicuous pattern here that they have a characteristic spacing, right? And if we zoom in to the outer layer, we see that it forms much like a roof, right? So this outer layer in teal is layered on top. It has these ridges at the cell-cell interfaces, these really characteristic ridges that are actually quite useful and highly conserved. It's how I actually did this whole data set in the first place. And also there's subcellular texture happening on top of the actual apical surfaces of the cells too. That is real, and that you can also find in the scanning electron micrographs too. So just one thing you can do in the lab, which you can't do in any other species usually, is you can learn stuff about where the proteins are coming from. So I'm going to just tell you right now that this eggshell ECM is assembled super locally. So this is an image of an eggshell that was produced by a completely wild-type epithelium. But only a subset of those cells are expressing the transgenic version. Okay, see if you can guess which cells are the ones who express it. Okay, so this set of cells can express it, and the fluorescent protein is basically moving nowhere. It's just assembling right at the surfaces of the cells. So that has motivated me to then pay close attention to what the shape of these cells are at the surface. Because if we want to understand how they make this structure, we would need to know actually the shape and secretory pattern of a single cell. Okay, so to do this very briefly, this is my interpretation. To do this very briefly, I did a screen. I'm not going to go into the weeds on this screen. I'm just going to show you the high-level results. But in brief, you can actually affect just how one gene works in one tissue in Drosophila. So it allows us to be crazy targeted like a scalpel to figure out how stuff works. So I targeted some cell processes that are implicated potentially in this process, and then identified some core genes in each one of these processes, picked a subset of those where there's really good evidence for how they work. And then you can increase or decrease the expression of those candidate genes just in this tissue, just while the eggshell is being made. Okay? And then afterwards, you can quantitatively characterize these altered ECMs in both of these two tagged lines. Then you can come up with more quantitative traits, more features, and associate those with genotypes and cell processes. Okay. I'm not going to name any of the genes specifically. I'll put them on the slide in case you're curious. So, all right, what are our big questions? Which processes can alter ECM structure, and what are the readily accessible axes of variation? So one, we can't actually change the size and shape of cells. Only one genotype did this. This is a gene that manipulated the stability of septate junctions. And you can see the area of these cell imprints has gotten way higher variance. This is a baffling result. I don't have much to say about it, but it's true that we can affect that one thing that tends to change a lot over evolutionary time. But that's the only condition that would do that. The rest of all the hits we found on the screen affect the subtle sort of like textural features. So we can characterize these ridges on the cells. You might imagine how we do that. You can come up with automated ways to identify these. then you can isolate that stuff, and then you can make estimates of how much protein has been enriched at these ridges, and then you can also measure things like their breadth and their thickness and that sort of thing. And when you do that, you find lots of examples. You find cases where you can add concentration of protein at these ridges. You can find cases where there's less protein at the ridges. Here's just some examples of how these distributions change over different genotypes. You can also detect changes in the 3D structure. So single genes expression changes can change the 3D structure. For instance, in this case, you can take lots of these ridges and you can project in Z and add them all together, and you can go from a nice crisp ridge in one case to a nice flat gene in another. So, we see ridges disappearing. We see the cell imprints disappearing over evolutionary time. I've always sort of wondered, how hard is it to get rid of these structures? And the answer is, you can change the expression of just one gene in just one tissue transiently, and that'll completely go away. So, okay, not that hard, I guess. And there's lots of ways to do it. That's the outer layer of the ECM. I'm also going to just briefly show you for this inner layer, these pillars, which it turns out are actually conserved across insects. There's these cavities in eggshells that relate to how much gas and water moves between them. So you can image these fluorescently. And then, of course, you can do normal CV stuff on these. You can identify where the pillars are. You can figure out who their neighbors are. You can measure stuff about how big they are and how well spaced they are. And then you can just characterize a bunch of different dimensions of this phenotypic diversity. And you can find lots of cases. Again, a single gene can turn these little puncta into worms for some reason. You can change how dense they are and how big they are. Here's just a few examples that show this sort of dramatically. So there's a lot of data here. And what we've learned is that these changes that we're having over evolutionary timescales, you could recapitulate in the lab just by taking one gene at a time, turning them up or down. I do not know if these are the things that evolution was changing, but we know it's at least plausible. And we've described what you might call the variational tendencies of the tissue. So I'm going to wrap up this by describing the data in a different way, which is all at once. Okay? So we have this high throughput phenotyping allows us to actually understand the broader structure of the phenotypic changes. So the columns here are morphological features that you can measure. The rows are experimental conditions. And in each one of these conditions, a single gene's expression level has been changed during eggshell formation. And I can just say that, I did not expect anything like this. What you typically expect for a screen is most of the genes you knock down or turn up do nothing. And then every once in a while, you get a cluster of them that all have the same phenotype, where A turns on B, turns on C, and they all have basically the same phenotype. And that phenotype is typically quite strong and easy to detect. That's like the whole basis of developmental genetics for the last 40 years. Instead, we saw something different, completely different, actually, which is that lots of these genotypes had subtly distinct, but distinguishable phenotypes that are multidimensional and subtle, right? So I think this is perhaps a less biased way of perturbing a developmental system. And I think what we've seen for lots of screens through history is that you're sort of intentionally looking for those really obvious unidimensional traits. So you're filtering your data for those that you can put on the cover of your paper submitted to Cell or whatever. But actually, if you were measuring lots of traits like this, you might find really subtle and complex and disorienting systems of developmental regulation. All right, so I think this gives us a nice foothold for doing interesting things in the future. And I'm gonna, let me think, what's the way to do this? I'm gonna skip this slide where I summarize, and I'm gonna give you 2 minutes for one more example case, a different structure and tissue altogether. And then I'll leave this on the screen when we do questions. Let's do it that way. So this other quick example is crawling locomotion, maybe closer to home for some of you guys. So, another thing you can do is to take these cell and tissue traits and use them to better understand locomotory behaviors or how those things relate to the ecological niche. And this is a project I was doing in collaboration with the lab of Ellie Heckscher at the University of Chicago. And they work on larval crawling phenotypes and also the neuromuscular control of those sorts of movements. So for a particular group of insects called Holometabula, there's this single origin of this worm-like body form. You might also know it as a caterpillar or a maggot, depending on the group. And they have these fascinating locomotory traits. And the basic movement is like a peristaltic wave that goes segment by segment by segment. And it passes down the body. And when it does, something interesting happens, which is that they grip onto the surface with something that's analogous to feet. They have these surface structures repeated, called denticle belts. You might have heard about this from the work of Eric Wieschaus or Christian Nusslein-Volhard, where they did a mutant screen and found that these little hooks got rearranged in different directions, and people learned like segment polarity genes, that kind of stuff. But it's just a sort of stereotyped surface pattern. Each one of these hooks is made from a single cell in the embryo, and then later you might have one cell making several of them. And if you zoom in on these things, they're really cool. They're these fine-scale cuticle structures. They all have sort of particular arrangements and shapes and polarities to them. And this long-standing hypothesis is that these cuticle outgrowths play a role in larval locomotion, mediated by what they're crawling on, right? So these interact with what you crawl on, and as you crawl on different stuff, you might need to grow different ways to grip the world. So we focused on Diptera because they are particularly delightful when you look at these outgrowths. Here's just one example drawing. And I think you can probably guess where I'm going next, which is that entomologists have documented larval morphology for a lot of species. And you can go through the literature and you can start to extract this stuff. And you can find textual descriptions of it, SEMs, drawings, all kinds of micrographs. And then you can take these and make this big data set where you've leveraged the thing which is scarce, right? Human expertise. And that's what we're kind of talking about all the time, whether you have people dropping key points or people identifying bears with a particular nickname or people who are scoring your cells. This is a particular type of expertise that's incredibly rare because this is done by taxonomists who might be one of only 3 people in the world who can identify and key out these larvae to species. And they're the ones who are doing the descriptive work. So we want to take their work seriously. In fact, we extracted, in that case, all the text descriptions of those phenotypes, which was a real mess, I'll tell you that. But what we can get is a general body morphospace for dipteran larvae, which is kind of cool. And then also, you can do stuff like pick out classes of these cuticle outgrowths and then plot their presence and absence across lots and lots of families, right? And so these are denticles on their thorax. There's also these hairs. We actually do it by body segment, so you get information on a segmentally specific way. You can also get these things called welts, which are little like grippers that they use to grip onto stuff when they're in the stream and the current is threatening to take them away. Anyway, this is ongoing. But if you have anything you think we should study with these data sets, please let me know. And with that, I'm going to do a little bonus summary, which is that these complex multidimensional changes to morphology can be coarse-grained in this one particular way, right? As changes to a relatively small number of cellular traits and the processes that underlie them. And my hypothesis is that those processes constrain and enable big macroevolutionary changes to this organism scale morphology that we all care about. And now I think we're finally amped, to maybe write that lit parser code I talked about, and to start assembling these image data sets from all this published work, because this stuff is extremely rare and very precious, and we should try to leverage the most that we can out of it. And conversely, as you do your work, I encourage you to be a good ancestor to the future version of me, because some of these people were much better than others, right? The number of people who didn't put a scale bar on their images drives me nuts. And so these are some of the people who contributed to these projects. And there's I worked in 2 labs here, and I've also been collaborating with a lab at the Field Museum. And then here's that summary of the eggshell stuff, which I'll leave up here when we do questions. But basically, I think this is a great system for the cellular basis of morphological evolution. Tons of these traits have convergently evolved, which I think gives us an opportunity to understand how cell stuff gives rise to tissue stuff. And you can recapitulate some of these in Drosophila in this really simple way by changing the expression of one gene at a time. And then there's these multidimensional and subtle morphological changes that result from, kind of bizarrely simple genetic perturbations. Okay. So with that, I'm happy to take questions. Yeah, let's look at maybe 10 minutes, 12 minutes? Okay.